Fundamental unit of life, video number 5. Students, in my previous video I had discussed about some of the cell organelles like endoplasmic reticulum, Golgi bodies and lysosomes. In this video I will be telling you about mitochondria, plastids and vacuoles. The features and functions of these organelles will be discussed, and at the end we'll be discussing about the difference between plant cell and animal cell, and also why the cell is called as the structural and functional unit of life. At the end of the video there is a small assignment that you have to do in your bio. Mitochondria are the rod-shaped membrane-bound organelles that are involved in oxidation of food and release of energy. Due to this function of mitochondria, the mitochondria is called as the powerhouse of the cell. Now, mitochondria are present both in plant as well as in animal cells. Let's have a look at the structure of mitochondria in detail. Now, each mitochondria is bounded by two membranes. This is the outer membrane of mitochondria, which is smooth and porous, and the role of this is to regulate the exchange of material between the cell cytoplasm and the interior of the mitochondria. Right. Now the inner membrane of mitochondria, have a look at the diagram here. The inner membrane of the mitochondria is highly folded. Like the outer membrane is smooth and porous, right? But the inner membrane is highly folded into finger-like projections. Look at the diagram that I am drawing, right? So the inner membrane, outer membrane, you see it's smooth and porous, whereas the inner membrane is highly folded into these finger-like projections towards inside. And these finger-like projections that you see, they are called as the cristae, right? Now this folding of the inner membrane into cristae increases the total surface area for ATP generating biochemical reactions. Because of these cristae that you see, these foldings that you see, they are the surfaces, or that's a membrane folding which has got those ATP generating particles called as the F1 particles, over which ATP, that is adenosine triphosphate, okay, that's called as the energy currency of the cell. Mitochondria is a site of oxidation of food. It's a place where food is oxidized and then the energy will be released. This energy that is released is stored in the form of ATP, right? And the ATP are generated over this cristae. They are generated over the cristae. So presence of these cristae or the infoldings of the inner membrane increases the total surface area for ATP generating biochemical reactions. Okay. And not just the mitochondria, the plastids are also bounded by two membrane, about which you will be learning. And the organelles that I have discussed earlier, that is the endoplasmic reticulum, the Golgi bodies and the lysosomes, those organelles are bounded by one membrane, but mitochondria and plastids, they are bounded by two membrane. Now, other important thing about the mitochondria is that the inner membrane of the mitochondria encloses a fluid filled region. There is a fluid filled area which is called as the matrix. Okay, this fluid filled area is called as matrix, and matrix is a place which contain enzymes. It contains enzymes that are required for oxidation of food. Like what happens is that once you have eaten food, or when the food is taken into the body and it is digested, finally the food that is digested food, that is glucose, will be taken into the cell, right? And once it is taken into the cell, there it has to undergo oxidation. It has to undergo oxidation, right? And this oxidation of food, glucose, a part of it occurs in the cytoplasm, about which you will be learning in your class 10, and complete oxidation of it occurs in the mitochondria. And for this oxidation, enzymes are required. Okay, and these enzymes are present in the matrix of the mitochondria. The matrix has the enzymes. The matrix of the mitochondria, that is the fluid that is present in mitochondria has the enzymes for oxidation of food and release of energy. One more very important thing about mitochondria is that the mitochondria's matrix also has DNA. There are DNA, that is, it has its own genetic material. DNA I had told you, deoxyribonucleic acid, that's a genetic material of a cell, and that's present in the nucleus. But apart from nucleus, it is also present in mitochondria and the plastids. And due to this presence of DNA, that is a genetic material, the mitochondria can divide on its own, one thing, and the other thing is because of DNA and also because of ribosomes. Ribosomes, you've learned that the ribosomes are present in the cell cytoplasm. The ribosomes, they are present in the cytoplasm, or they are present attached to the rough endoplasmic reticulum, and the ribosomes, they are the sites of protein synthesis. So ribosomes are also found in the mitochondria, and apart from the ribosomes that are found in the mitochondria has DNA. There are circular DNA as shown in this picture. There are DNA present in the mitochondria, and due to the presence of DNA, the mitochondria can divide on its own. That's one thing. Another thing is that the mitochondria, because of the presence of DNA, can prepare some of its own proteins and lipids. So it can make its own proteins and lipids due to the presence of DNA, and for those, thus they are not dependent totally upon the nucleus. Because of this property of mitochondria, it is having its own genetic material, due to this, mitochondria is also referred to as a semi-autonomous organelle. This is called as a semi-autonomous organelle. Now, so just let us just summarize what I have told you. The mitochondria is bounded by two membranes. The outer membrane is smooth and porous, while the inner membrane is folded inwards into projections which are called as cristae, and they increase the surface area for ATP generating biochemical reaction. Next important thing about mitochondria is that the inner membrane of mitochondria encloses a fluid filled matrix, and this contains the enzymes that are required for complete oxidation of food and release of energy, right? So oxidation of food occurs in the matrix of mitochondria, and the energy that is released during the process will be used up for making ATP. It's used up for making ATP, and this ATP is generated over the cristae of mitochondria, over this finger-like projections that you see, which are called as a cristae. So over the cristae of the mitochondria, this ATP is generated. And other important thing that I had told you about the structure, that the mitochondria contains its own genetic material and ribosomes, and because of the presence of this DNA and ribosomes in the mitochondria, this organelle can prepare some of its own protein. The organelle can prepare some of its own protein. Now learn about the functions of mitochondria. First important thing and the most important function of mitochondria is that it is a site of oxidation of food and release of energy. Because of this property of mitochondria or function of mitochondria, it's called as a powerhouse of the cell. Now, as stated to you that the mitochondria, the matrix of the inside the matrix of the mitochondria, the food is completely oxidized. It is burned, and during the process, energy is released. You have already learned in class 8 that whenever combustion occurs, whenever the materials burn, energy is released. So when food is burnt or oxidized in the matrix of the mitochondria, energy will be released, and that energy that is released will not be used up immediately. It will be used to make ATP. In other words, we can say that the energy that is released during oxidation will be stored in the form of ATP, and this ATP stands for adenosine triphosphate. Okay, it stands for adenosine triphosphate. So in other words, we can say that the energy that is released during oxidation of food, is stored in the form of ATP, that is adenosine triphosphate, and this ATP will be used in various metabolic activities for synthesis of many biochemicals, for the mechanical work to be performed, like any kind of activity. If we take ourselves as an example, for each and every work that we do, for the movement of muscles, for the neural conduction, and for many other activities that you perform. Even when you are sleeping, it's not that when you are sleeping your energy is not required. Energy will be required all the time, because even if you sleep, your organs are functioning, and for their functioning, for their working, energy will be required. So this energy is obtained from ATP. ATP will store the energy, and that will be used up whenever they are needed, okay, for synthesis of different biomolecules, that is for making proteins, for amino acids, or for any other purpose, synthesis of any kind of biochemicals. The energy will be obtained from ATP, or for my various mechanical activities, this energy will be obtained. So here this is a diagram that shows how ATP is used up. Though this diagram which is shown over here is not required for class 9, but for a better explanation this will be helpful to you. ATP stands for adenosine triphosphate, and when this adenosine triphosphate, it stores energy, okay, it stores energy in it. Whenever energy is required, this ATP will undergo breakdown, okay, it will be breaking down into ADP, that is adenosine diphosphate, and inorganic phosphate. And this, during this breakdown, this will happen whenever you need energy. So when this breaks down, then the energy that was stored in ATP will be released, and this energy that is released during the breakdown process, this energy that is released will be used for performing various mechanical activities, or will be used for synthesis of various biochemicals, okay. So these are the important functions that are performed by the mitochondria, okay. Now learn about the plastids, which are the organelles that are found only in plant cells. The plastids are disc-shaped or lens-shaped membrane-bound organelles that are found only in plant cells, and they are specialized to manufacture food and to store food, okay. So here this diagram of the plant cell shows, the diagram that you see here shows various organelles. You find mitochondria in plant cells as well as in animal cells, but these plastids here, the chloroplasts are being shown. So the plastids you find only in case of the plant cells, and these plastids, as I said you, they are specialized for manufacture of food and for storing food. Now plastids are of two types. There are two types of plastids, the chromoplast and the leucoplast, okay. The colored plastid, that is the plastids which have pigment in them, they are called as the chromoplast, and those plastids which are colorless, that is they don't have any pigment present, they are called as the leucoplast, okay. Now coming to the chromoplasts are of two types. These chromoplasts, there are of two types. The two types of chromoplasts are the chloroplasts and the non-green chromoplasts. The green chromoplasts, they are called as a chloroplasts. These green chromoplasts are known as the chloroplasts, which you all are aware of, and these chloroplasts are the green plastids because they have got green pigment known as chlorophyll, and because of the presence of chlorophyll, the chloroplast becomes the site of photosynthesis. The role of chlorophyll in the process of photosynthesis is to trap light energy. It traps the light energy that is required for photosynthesis, right? And the other type of chromoplasts are the non-green chromoplasts. These non-green chromoplasts have got red, orange and yellow pigments, okay. So the non-green chromoplasts, they have got red, orange and yellow pigments, and the function of these chromoplasts is that they give color to the flowers and fruits. The tomatoes, they are red in color, or the yellow color of the petals of the flowers, or various colors are because of the presence of chromoplasts in them. And because of the chromoplasts, this chromoplasts in these fruits and flowers, they help in pollination and dispersal of fruits and seeds, right? The third kind of plastid that you find are the leucoplasts, okay. These are colorless plastids because they don't have any pigment in them, right? And the function of leucoplast is that the leucoplast is specialized to store food in the form of starch, proteins and fat, okay. So they are specialized for storing starch, food, proteins and fat. A potato tuber, when it contains the leucoplast that stores the starch, Let us now study about the detailed structure of a chloroplast. So this is the structure of a chloroplast, and like the mitochondria, you find the chloroplast is also bounded by two membranes, the outer membrane and the inner membrane. So like mitochondria is bounded by two membranes, and the chloroplasts are also, in fact all the plastids are bounded by two membranes, the outer membrane and the inner membrane. The inner membrane encloses a fluid filled region which is called as the stroma. So here you see there is a fluid filled region which is called a stroma present in the inner membrane, that is enclosed by the inner membrane, right? And this stroma, in the stroma floats many membrane bound flattened sac like structure. Have a look at this diagram. You find there are flattened sac like structures. Okay, flattened sac like structures which are stacked one upon the other like a stack of coin. Okay, so each flattened sac like structure is called as the thylakoid, and a stack of thylakoid is called as granum. A stack of thylakoid is referred to as granum. Now, significance of thylakoid is that the membranes of the thylakoid, they contain the chlorophyll pigment, and so it is the thylakoid that is a site for where light energy is absorbed. Now, the stroma of the chloroplast contains enzymes that are required for converting carbon dioxide to carbohydrate. What happens in photosynthesis? It is in photosynthesis, it is carbon dioxide and water that combines to form carbohydrate. So, this conversion of carbon dioxide to carbohydrate will occur in the stroma, and for that energy that is required will be obtained from light, and that light energy will be trapped by the chlorophyll molecule. Then there ATP is generated, about which you'll be learning in your senior classes, and that energy that is generated will be used to convert carbon dioxide to carbohydrate. And that's why you find in this chloroplast, you'll also find, because carbohydrates, they will be made in the form of glucose. Food is made in the form of glucose and immediately stored in the form of starch granules. So in the chloroplast you also find starch granules too. Then one thing common that you find in case of chloroplast in the mitochondria is that, like the mitochondria, the chloroplast also has its own DNA and ribosomes. There are DNA and ribosomes and DNA present in chloroplast also. And since the chloroplast contains its own genetic material, that is DNA, like mitochondria, it can prepare its own protein. Okay, so it's both mitochondria and chloroplast. They are the two organelles that have their own genetic material, right? So these are the important points. This slide, just have a look at this important points. These are the important points that you need to note. First is that each chloroplast is bounded by two membranes. Then inside the chloroplast are the stacks. Chloroplasts are stacks of membrane-bound flat sac-like structures called as the thylakoids, which contain chlorophyll that traps the light energy, and the chloroplast encloses a dense fluid which is called as stroma. The stroma contains the enzymes that are required for converting carbon dioxide to carbohydrate, the process that happens in photosynthesis. And finally, the most important thing, like the mitochondria, the chloroplast has its own DNA and ribosomes, because of which it can prepare its own proteins. Amino acids, proteins and lipids can be prepared, which may be used for making of the chloroplast membrane, right? So now here this diagram or this slide shows the similarities and dissimilarities between mitochondria and the chloroplast. So have a proper look at this. Since I have already discussed about the two organelles, let's just have a look at this similarity and dissimilarity. The similarity between mitochondria and chloroplast is that in both have double membrane structure. That is one thing. Both of these have their own DNA and ribosomes. Okay. From the class 9 point of view, these 2 points you need to know, that both chloroplast and mitochondria, they are double membrane organelles and they have their own DNA and ribosomes, right? Other important, the differences between the two organelles is that mitochondria is rod shaped, whereas chloroplast is lens shaped. Then Mitochondria contains cristae, those finger-like projections or the folds of the inner membrane, whereas the chloroplast, they contain stromal thylakoids. I said you they are flattened sac-like structures which arrange as grana. So there are thylakoids and grana present, whereas mitochondria has got cristae. Then the other difference that you find is that the mitochondria does not contain photosynthetic pigments, that is chlorophyll, which is present in case of chloroplast. Next important thing that you see is that the mitochondria are found in all animal and plant cells. Every eukaryotic cell will have mitochondria, right? But chloroplasts are found only in plant cell, right? That's another difference. Now, functional difference you find is the mitochondria is involved in aerobic respiration. It is a site of oxidation of food which occurs in presence of oxygen, right? But chloroplast, that is involved in photosynthesis. So these are the similarities and differences between mitochondria and the chloroplast. Now learn about the vacuoles. Vacuoles are membrane bound sac-like structures. These are membrane bound sac-like structures which contain solid and liquid substances. Now both plants and animal cells have vacuoles. But the difference that you find is that in case of plants, there is a central vacuole present. There is a central vacuole present which occupies 50 to 90% of the cell volume. When the plant cell is young, the vacuole will be smaller in size. But as the plant cell matures, the center of the cell gets occupied by a central vacuole, which is also called as the sap vacuole. Okay, there will be one or two large central vacuole occupying like more than 50 to 90% of the cell volume, and because of the central vacuole, the nucleus in the plants is pushed towards the side or periphery, right? Now, in case of animal, just have a look at the animal's vacuole. In case of animal cells, the vacuoles are quite small in size. The vacuoles are very small in size. Now, the vacuole that is present in the plant cell, this vacuole of the plant cell has got a membrane, and this membrane it is a selectively permeable membrane, and it is called as the tonoplast. And the fluid that is present in this vacuole is called as the cell sap. Okay. Try to recall, when I was discussing about the osmotic solutions, there I had told you that the plant cells, they take water by endosmosis and they may lose water by exosmosis, right? And when does this happen? You have learned that if the plant cell is present in a hypotonic solution, that is a solution whose osmotic concentration, whose water concentration is higher, it has high water content when compared to what? When compared to the cell sap. If the cell sap which is present has got low water content and outside there is more water, then the cells are going to take in water, right? So that this clearly shows that the concentration of the cell sap is very much important. If the water concentration of the sap is lower than the outer concentration, in that case the cell is going to gain water by endosmosis, right? And if the water concentration of the cell sap is higher, if the cell sap, which is a fluid in the vacuole, is higher than that of the outer solution, then in that case the plant cells are going to lose water by exosmosis. Okay, they are going to lose water by exosmosis. This will happen and eventually the cell will get plasmolized. So this concentration of the cell sap determines whether the plant cells are going to gain water or are going to lose water, right? Now let us discuss about the different types of vacuoles. Vacuoles are of three types, right? There is the sap vacuole, then I will be telling you about the food vacuole, which you might be aware of, and there's another kind of vacuole which is called as a contractile vacuole. So let us first know about the sap vacuole or the central vacuole. As just now I've told you that the sap vacuole is a fluid filled sac which is occupies center of the cell, and this sap vacuole, it has got a liquid which is called as a cell sap. This cell sap contains many dissolved substances like sugars, minerals, salts and amino acids, and recently like some lysosomal enzymes, like plant cells do not have lysosomes, but some lysosomal enzymes have been detected in this sap vacuole, in the sap of the plant cells, right? Another thing is that the plant cells sap vacuole or the central vacuole also stores waste materials. They also store waste material. So here the sap vacuole, it stores many of the important substances that are required for the survival of the plants, like amino acids, sugars, organic acids, proteins are stored. This is one important function of the sap vacuole. The sap vacuole also stores, sap vacuole or central vacuole also stores the waste products. That is also because plants do not have any specialized excretory structures or organs. So what they do is they store the waste products in the living cells, will be storing the waste products in the inside the sap vacuole. And because of the tonoplast, the outer membrane that the vacuoles are having, it's a selectively permeable membrane. So this membrane will not allow the waste to move out, and so the activity of the cell is not affected. Other important function of the sap vacuole is that the solutes present in the sap vacuole maintains the proper osmotic concentration of pressure of the cell, which is important for providing rigidity and turgidity. Like how is the rigidity and turgidity obtained? The osmotic gain of water. It is gain of water. When this will happen when the sap vacuole has got lower water content than outside. So when the water enters the sap vacuole, the vacuole stretches, and that causes a protoplasm to stretch, and thus the cell becomes turgid and rigid. So turgidity and rigidity is maintained because of the osmotic concentration of the sap vacuole, or the osmotic concentration of the sap present in the sap vacuole. So these are the important functions of the sap vacuole. Now let us know about the next type of vacuole, which is the food vacuole. Not in detail you require. Food vacuoles are found in protozoans like amoeba. In case of amoeba, as already discussed, I have already discussed about amoeba in previous videos also. So in amoeba, like once the food is ingested, have a look at this diagram of amoeba. So once the food particle is taken in, can you recall by which process the food particle is taken in? It is endocytosis. It's called as endocytosis. So when they take in food particle, what happens is that the particle will be taken inside by making a cup-shaped structure, that's endocytosis, and then the food particle will move into a sac-like structure which is called as the food vacuole. Then I had told you that the lysosomes which are present in the cell, they fuse with this food vacuole and digestion of food occurs. So in this way, this food vacuole which is present in these protozoans or some protists, they help in digestion of food. They are the site where the food is completely digested with the help of digestive enzymes that is secreted by the that is produced by the lysosome. So it's the lysosome that produce the lysosomes. They have the digestive enzymes, and these lysosomes fuse with the food vacuole, and then intracellular digestion of food, that is digestion of food inside the cell, occurs in the food vacuole. So that is another kind of vacuole which is present in some of these organisms like the protozoans, like amoeba, right? Then there is third kind of vacuole which is called as the contractile vacuole, right? Contractile vacuoles are found in some protozoans like amoeba, like in case of some unicellular algae, you find this, the paramecium also has this contractile vacuole, and the role of contractile vacuole is it helps in expelling excess of water. Okay, these protozoans which live in fresh water, mostly they live in fresh water, these contractile vacuoles will help in removing the excess water. This diagram shows how water Not just the water, waste materials also are expelled. So in this diagram, as you can see, what happens is that since these animals, when they are there in water, outside there is water, so definitely water is going to enter. Look here, the water is entering. So as water enters, it will enter into these vacuoles, which are called as the contractile vacuole. And as the water enters the contractile vacuole expands. And finally what happens is these contractile vacuoles will move towards the side of the cell, and when they come in contact with the plasma membrane, when it comes in contact with the plasma membrane, the contractile vacuole contracts suddenly, and then they expel all the water that has entered the cell along with the waste material to exterior. So in this way, the contractile vacuoles, they help in removing excess water and waste materials in those protozoans and unicellular algae, which are mostly found in fresh water. So that's about the 3 types of vacuoles. And then now let us learn about, like, since I have already discussed about the plant cells and animal cells, so have a quick look at the difference between the plant cells and the animal cells. How are the plant cells different from the animal cell? Now plant cells, as you all know, they are larger in size when compared to that of the animal cells. Then in case of plant cells, as already discussed, the plant cells, they have got a cell wall. In a plant cell, you find there is an outer rigid covering which is present, that is called as the cell wall. But this cell wall is not present in case of animal cell. In case of animal cell, the outermost covering of the animal cell is the cell membrane. So cell membrane is found, cell wall is present in plant cells, whereas the cell wall is absent in case of animal cells. Then the third difference that you find in case of plant cells and in animal cell is that the plant cells, they have got plastids. Okay, now we have discussed in this video, I had told you about the plastids. So plant cells have plastids like chloroplast, chromoplast, leucoplast, but plastids are absent in case of animal cells. Animal cells don't have plastids. Then the next difference that you find is that in case of plant cells, the Golgi bodies are not well developed. The plant cells do not have well defined Golgi bodies. There are subunits of Golgi bodies scattered in the cytoplasm, and these subunits of Golgi bodies which are present in the plant cells performing the same function that was discussed, these subunits of Golgi bodies are called as dictyosomes. Okay, they are called as a dictyosome. So the Golgi bodies are scattered in the cytoplasm of the plant cells, but Golgi bodies, a complete unit of Golgi body, is present in animal cells near the nuclear envelope and the rough endoplasmic reticulum. The next difference that you find in case of plant cells and animal cell is that the plant cells do not have centrosome and centriole. These centrosome and centrioles, they take part in cell division. Okay, the centrosome and centriole, here there is a centrosome present in animal cells which has an important role to play in cell division. So these centrosomes or centrioles are present in plant cells, but are present in animal cells, but these are absent in case of plant cells. The next thing that you find is that the vacuoles, and just now I have told you that large size vacuoles but few in number are present in case of plant cells, but in case of animal cells, the vacuoles are very small in size and they are many in number. They will be present in many small vacuoles will be present in case of animal cells, right? Now next difference, so here this is about, and then the position of the nucleus in case of plants and animal cell. In case of plant cell, the nucleus is present at a side. Why? Because there is a central large vacuole. So because of the vacuoles present, the nucleus is present at the periphery, at the side of the plant cell. But in case of animal cell, you find that the nucleus in case of an animal cell is centrally placed. It is present in the central position. So these are the differences. The differences are shown in this slide. So these are the differences between the plant cells and the animal cells. So this is the diagram of a plant cell. Just have a look at the different parts of the plant cell. You've got the nucleus, structure of nucleus is already discussed. Then near the nucleus you've got the Golgi apparatus. I've already told you the function of it. Then you also find that the plant cells, they have got an outer rigid covering of cell wall which is absent in animal cells. Then in case of plant cells, you have and as well as in animal cells, you will find rough endoplasmic reticulum, there is smooth endoplasmic reticulum, there is cytoplasm present. Okay, lysosomes, this should be removed. So lysosomes are not present in plant cells. And then you also find that mitochondria, ribosomes, these are all present in animal cells also. So this diagram this diagram that is shown over here is a diagram of plant cell that's given in your NCERT book, and the next is the diagram of animal cell. Just have a look at the diagram of animal cell that's given in your NCERT also. So let us see the parts of the animal cell. You've got the plasma membrane, then here is a centriole. Just now when I was discussing about the difference, I said you that the centrioles are present only in animal cells, right? So very close to the nucleus you find two microcylindrical structures, right? These microcylindrical structures are called as centrioles, and centrioles are present in an area that's called as a centrosome. So centrosome is found in case of animal cells, but it is absent in case of the plant cells. Then other important structures that you see is you find lysosomes, the digestive bags or societal bags of the cell. Then you find the ribosomes. The role of ribosome is to synthesize protein. It is a site of protein synthesis. So ribosomes, these ribosomes that you see, you find them either attached to the rough endoplasmic reticulum. Over here it is attached to the rough endoplasmic reticulum, or they may be lying free in the cytoplasm also. So those ribosomes, they are not surrounded by any membrane, and these ribosomes, they are the site where proteins are synthesized. Then like in case of plant cells, in animal cells also you find this mitochondria present. Mitochondria, structure of which I have already discussed, it's called as a powerhouse of the cells. So this is present in plant as well as animal cell. And what else? You find the rough endoplasmic reticulum, the smooth endoplasmic reticulum found in animal cells as well as in plant cells. And then here is the Golgi body or the Golgi apparatus, which is a complex unit in case of animal cell, which is shown over here, and it is present as subunits scattered in the cytoplasm as dictyosome in case of plant cells. So this diagram is a diagram of animal cell. So here let us, this is a flow chart showing the different parts of a cell. Okay, so let us summarize the entire of it. The cell has got three functional regions, that is the cell membrane, the cytoplasm and the nucleus, right? And the nucleus. Now the cell membrane, as already stated, it is a selectively permeable membrane. We have discussed how transportation of materials occur in and out of the cell, right? Now cytoplasm, as I said to you, the material which is enclosed between the cell membrane and the nuclear membrane is called a cytoplasm, right? So the cytoplasm contains cell organelles and cell inclusions. So cell organelles that are present are endoplasmic reticulum, the mitochondria, the Golgi apparatus, ribosomes, right, lysosomes, centrosome and plastids. And of these organelles which are present, the endoplasmic reticulum, the Golgi apparatus and the lysosomes, they are bounded by single membrane, whereas the plastids and the mitochondria is surrounded by two membranes. Then what else I said you? The centrosome and the ribosomes, they are not surrounded by any membrane. They are not surrounded by any membrane. So now for now you are learning it as organelles, these as membraneless organelles, but if we go with the definition of organelle, we define it as membrane bound cytoplasmic structure. So this may not be considered actually as an organelle, right? Then there are cell inclusions. They include the granules which are present in the scattered in the cytoplasm and the vacuoles about which just now I have told you. Now nucleus has got nuclear membrane, the nucleoplasm, the nucleolus and the chromatin. The chromatin contains DNA and histone proteins. It's a chromatin which condenses and forms the chromosome, right, about which I have discussed in detail. So the cytoplasm is the material between the cell membrane and the nucleus, right, and the nucleus, and the cytoplasm along with the nucleus, what is it called as? It is called as the protoplasm, right? and every component of the cell is quite important. Each and every organelle of the cell is very important. They have an important role to play, and so a cell without any organelle will not be able to perform properly. And these organelles are common to all kinds of cells, irrespective of the shape of the cell in which organism the cell has been taken. All living cells will have the same basic structure, and every living organism is made up of cells and the cell units, right? And that's why the cell is called as a structural unit of life, right? So here lastly, let us know, why is a cell called as a structural and functional unit of life? A cell is capable of performing all life activities such as excretion, respiration, and hence it is called as a functional unit of life. And the cell is the smallest unit of life of all living organism. Every living organism, whether it is a single cell organism or a multicellular organism, a small or a large organism, all organisms are made up of cell, and hence the cell is called as a structural unit of life. And each cell, thus it acquires its structure and ability to function because of the organization of membranes and organelles. The membranes, they have got a very important role to play, and every organelle has got a specific function to perform. A cell without a particular organelle will not be able to work for a longer period of time, right? So because of all these reasons, the cell is called as the structural and functional unit of life. Lastly, let us, on the basis of the video that you have watched just now, this is an assignment that is to be completed in your copy. So the first question is, why is the mitochondria called the powerhouse of the cell? Then discuss the similarities and dissimilarities between mitochondria and chloroplasts. Give a comparative account on different types of plastids. How do vacuoles maintain turgidity of the plant cells, and differentiate between plant cell and animal cell? And what are vacuoles? Write their functions.